Hello. Hello. Good evening. Good evening. Good evening. Hello. Can you hear me? If I'm audible, then you can give me a thumbs, okay? Hello. Good evening to all. Welcome to Go Score Learning live session. We are the fastest growing edtech in GCC. We make learning easy for our students. So here, dear students, myself Amala Anjos, and I'm here with you to take the new chapter of a biology textbook, that is molecular basis of inheritance. Okay, so while we are discussing about the different distribution of marks of different chapters, you come to know that it is very important to study this chapter and the previous chapter that we have already discussed. Okay, so let's see what are the different topics that we have to study in this particular chapter. Okay, so if you are looking into the chapter, first we have to study about the nucleic acid, that is DNA. Along with this DNA, we have to learn about RNA too. Okay, then after that we have to come across the structure of DNA. Structure of DNA, how the DNA is made up of. How DNA which is present in our body is made up of. Okay, then next one, it is about packaging of DNA helix. Okay, how DNA helix is being formed. Okay, how DNA will be wrapped inside the nucleus and how it will be formed. Okay, so next one, it is transforming principle. Transforming principle is coming under how this genetic material has been discovered. Okay, from in or inside that particular topic, we have to discuss about how the DNA was found out in the cell or in the different organism. Okay, then we have to study about biochemical characterization. Then next we have to study about Hershey's experiment to prove DNA as the genetic material. Then about properties of genetic material, that is about DNA and about RNA. Next will be in detail we have to study about RNA. After studying about RNA, we'll be coming to the next topic, that is replication. Replication in sense how the DNA is replicating to form the another copy. Okay, in after that we have to study in replication how the replication is taking place. It has also proved with the help of an experiment, and that experiment is actually Michelson's and Star experiment. It is very important that you have to learn. All the experiments that is inside this chapter is very important. Usually, the CBSE have a tendency to ask the questions from these experiments. So, it is very important to study the different experiments. It is easy to study. Okay, will make you to learn easily. Okay, then next after that we have to learn about transcription and translation. So if you are considering transcription, after transcription you have to learn about genetic code, then about tRNA, different types of RNA we have to learn. If you are considering RNA, we'll come to know that there are three different types of RNA, that is mRNA, tRNA and rRNA. So in this chapter we'll be in detail we'll be looking into every types of RNA and the function of each RNA at particular phases. So if you are considering transcription, different types of RNAs are included in that. Okay, then after that transcription, we will be studying with translation. In translation also, the different types of RNA has to be present. So we have to understand what is the basic function of different types of RNA. Then we will be coming across translation. Okay, translation. The three important process that we have to learn in this chapter is replication, transcription and translation. Okay, so the translation will be at the end of this chapter. It is about protein making, how the proteins are making in our body, how these proteins are helping in different functions of our body. Okay, so after this we will be coming into regulation of genes, how different genes are regulated in our, not in our body, in other organisms. Okay, we'll be taking an example from other organism to learn how transcriptional or how translational modifications will be coming, how it will be regulating. For that, we'll be studying about lac operon, which we'll call it as lactose operon. Okay, when lactose is present, this lac operon will be functional. Okay, then different types of operons are present or different types of regulations are being studied. For example, we can say Tryptophan operon is another kind of operon which functions in other organisms. Okay, so there are different mechanisms that we have to come across. Among that, lycopron is one of the important topic that we have to study in this chapter. Okay, usually I told you the questions will be asking from this particular topic also, lycopron. It is very, very, very important. Okay, so next it is DNA fingerprinting. It is quite interesting topic. After studying every process, after studying every experiment, we have to study how DNA fingerprinting will be taking place. Okay, you know that if there is any dispute in parental determination, then or usually the people will go for DNA fingerprinting. And also, if there is any crime scene, if we have seen in different movies that right, we'll go for DNA fingerprinting. So, what is actually DNA fingerprinting? We'll be discussing in our session what is DNA fingerprinting and what are the different features of DNA fingerprinting with examples, or how we will be analyzing. We will be studying that in the coming classes. Okay. So after that, the last one we have to learn about human gene project. Dear student, what is human genome? See, what are the different genes which is present in our body? See, if you're considering our chromosomes, there are 23 pairs of chromosomes, right? Each chromosome is having different genes. Each genes are characteristics. Each genes will give you different characteristics of your body. Your hair structure, your hair color, your eye color, your skin color. Everything is being determined by the genes which is present inside the chromosome. So, human genome project, if human genome project is studied or properly analyzed, everything can be sorted out. If different diseases or genetic diseases are being present, this genetic disorders can be uh, taken away, or it can be cured with the help of the sequences, analyzing the sequences for human genome. Okay, so this is also very important to study human genome project. Different genomic projects are going on now. If you people are interested, you just go through the scholars or what are different research papers, then you will come to know there are different researches that is going on in our society to understand what are the different genomic sequences which is present in different types of organisms. Okay, so if you're considering our genes and the monkeys, apes genes, almost the genes are similar. A few change is there inside the ape. That is why it is looking like ape and we human beings are looking like human beings. Okay, so these are the things that we have to come across in this chapter, and you have to be thorough with the topics that is coming under this chapter. First, be thorough with what are the different experiments coming under this chapter, then study about that. See, the biology is very important. You know that there are different terms in biology, right? So first learn the terms and then you modify the answer and study and develop a concept about that. Okay, so that's all about the general introduction of this chapter. Today we'll be discussing a simple topic, very simple topic, that is about nucleic acids. Okay, so we'll be moving to the class. First, as I have told, see the different things in our body. I told you that the skin color, we have studied in the previous chapter that there are some factors present in our in different organisms, right? There are some factors present. Okay, if you're considering there are some factors present inside the organism. So when Mendel was experimenting his experiments, he was not knowing that it was genes. Okay, he was never knowing that it was genes. But later on, the scientists worked on it and they came to a conclusion that it is gene. Okay, so they started finding out what is actually present inside the gene, and they found out what is the nucleic acid, or the genetic information that is carrying from one generation to next generation is DNA, and the other genetic material is RNA. So there are two different types of nucleic acid. If you're considering nucleic acids, okay, nucleic acid, we'll call it as nucleic acids. Okay, so if you are considering nucleic acids, what are the major nucleic acids that are present? That is DNA and RNA. Okay, DNA and RNA. So what is DNA? We'll write it as DNA as from very lower classes onwards. You know that DNA is deoxyribonucleic acid. Okay, deoxyribonucleic acid. Okay, deoxyribonucleic acid. Whereas in the case of RNA, we'll just write it as ribonucleic acid. Okay, so there are some differences in DNA and RNA. There are some differences. That's why we are calling it as DNA and RNA. If you're considering the eukaryotic organism and other prokaryotic organism, the DNA will be the genetic material. Okay, but if you are considering some viruses, the genetic material will be RNA. So we are considering both DNA and RNA as the genetic material or nucleic acids. Okay, so if we are considering DNA, we have to study the structure of both this nucleic acid, how these nucleic acids are made up of. Okay, how DNA is made up of, how RNA is made up of. So if you are considering the structure of DNA, if you are considering the structure of DNA, this DNA is actually made up of. Okay, so we are considering DNA and RNA, both DNA and RNA. If we are considering the structure of DNA and RNA, this structure is mainly composed of three components. Okay, first component is ribose sugar. Okay, what is the component? First component, it is ribose sugar. Okay. Okay, so what is the second component? Second component, if we are considering the second component, it will be nitrogen base. Okay, what it will be? It will be nitrogen bases. Okay, there are some nitrogen bases present inside. So if you are considering the third one, it is the phosphate group. Okay, so what are the different components present? Okay, what are the different components present? See, first one it is ribose sugar. Be thorough, make it thorough, okay? Ribose sugar is the first thing, and the second thing, it is nitrogen bases. Okay, what is nitrogen bases? It is high or rich in nitrogen. Okay, next one it is phosphate group. So these three are the basic components of DNA. Okay, and RNA. So let's see each and everything in detail. First, we will be looking into ribose sugar. So if you are considering ribose sugar, okay, so if you are considering ribose sugar, say you know how to draw the ribose sugar, right? So this will be the structure of ribose sugar. OH, H, then second carbon will be having H, OH, and third carbon will be also having H, OH, and the fourth carbon is attached with H, and it is attached to the fifth carbon, CH2OH. This is the structure of ribose sugar, right? So if you are considering RNA, okay, if you are considering RNA, we know the name as ribonucleic acid, right? Ribonucleic acid. So the ribose sugar present inside it, it will be ribose sugar itself. Okay, ribose sugar as itself. Okay, so if you are considering DNA, next one it is DNA, right? Next one it is DNA. So we have to consider both the condition. So if you are considering DNA, inside DNA also there is a kind of ribose sugar, but the name is deoxyribonucleic acid. So among one position, one oxygen will be eliminated. So in which position? Sorry, excuse me. In which position it will be eliminated? Okay, so first position OH, H will be present. In the second position it will be H, OH, and in third position also it will be H, OH. Okay, and at fourth position also, as I have said, it is connected with one hydrogen and then CH2OH. Okay, so this is the first carbon. Okay, so if you are considering the number of carbons, this will be the first carbon. Okay, this will be the first carbon, this will be the second carbon, and this will be the third carbon, and this is the fourth carbon, and this is the fifth carbon. Okay, so it is very important, dear students. It is very important. See, in the second carbon, you can see an OH. Okay, so the oxygen of the second carbon will be eliminated. Okay, oxygen of the second carbon will be eliminated. Okay, oxygen of the second carbon will be eliminated. So it will be known as deoxyribose. Okay, it will be known as deoxyribose. So you understand what is the difference between RNA and DNA? What is the difference of ribose sugar which is present in both DNA and RNA? Got it? So if you're considering RNA, we can say that it is a ribose sugar. If you're considering the DNA, the structure of DNA, the basic unit is deoxyribose, wherein the second position, the oxygen will be eliminated. Okay, clear. So this is the first component of DNA, that is ribose sugar. Not in DNA, both in DNA and RNA. Okay, so we will be moving into the next component, that is nitrogen bases. Okay, so next, what is the next component? It is nitrogen bases. So, as I have mentioned earlier, it is nitrogen bases. So what are nitrogen bases? I told you that nitrogen bases are actually rich in nitrogen. Okay, the presence of nitrogen will be more. Okay, so if you are considering nitrogen bases, the nitrogen bases can be classified into two groups, that is purines and pyrimidines. What are they known as? Purines and pyrimidines. So if you are considering purines, okay, purines, so they are classified into purines and pyrimidines. Okay, purines and pyrimidine. So if you are considering purines, if you are just considering purines, how these purines can be classified? These purines are again classified into two different groups or two different nitrogen bases, that is adenine. Okay, what are the two? Adenine. First one is adenine. Okay, second one is guanine. Okay, so if you are considering the nitrogen bases present in DNA and RNA, what are the purines? What are the purines present? It is adenine and guanine. So next one it is pyrimidine, right? So if you are considering pyrimidine, what are the two different pyrimidines present inside the DNA? It will be thymine and cytosine. Okay, thymine and cytosine. See, we have to learn new different names. Okay, new different names. So be thorough with the names. So if you're considering purines, adenine and guanine is the purine. If you're considering pyrimidids in DNA, it will be thymine and cytosine. Whereas in the case of RNA, see, this thymine is replaced by another one, that is uracil. Okay, so if you're considering DNA, thymine and cytosine is present. But if you're considering RNA, cytosine and uracil is present where thymine is absent. Okay, so these are the different nitrogen bases which is present inside the DNA and RNA. Once again, I'll be writing in a little more easier way, so it will be easy for you to understand. So see, if you are considering DNA. Okay, so this is DNA and sorry, one second. Okay, so these are the purines. Here we can write the purines. Here we can write the pyrimidines. Okay, so if you are considering DNA, what are the purines present? Okay, if you are considering the DNA, what are the purines present? First one it is adenine. Okay, second one it is guanine. Okay, so next, if you are considering the pyrimidines, what are the pyrimidines present? It is cytosine and thymine. Cytosine and thymine. Okay, so if you are considering next RNA, see, next one it is RNA. So what are the purines which is present in RNA? The same thing. There is no change. Okay, first one it is adenine. Okay. Then second one, it will be guanine. Okay, fine. Got it? So, in the case of pyramids, there is a change, right? In the case of pyramids, there is a change. So, we'll be writing in another color, that is cytosine. Cytosine will be present inside the RNA. Okay, instead of thymine, there is no thymine inside the RNA. But instead of thymine, what is present? What is present? Uracil is present. Uracil is present. Okay, uracil is present. So this you have to keep in remember. You have to keep in remember. You have to study this basic thing. Okay, if it is thorough, then it is easy to make notes on this and easy to score in your exams. Okay, so take a note on this. Okay, take a note on this and study this. Okay, so these are the different what we can say base pairs. Okay, these are the different nitrogen bases that is present in DNA. Okay, is it thorough? Is it clear? If it is clear, then you can give me some thumbs. Okay, I'm not getting any thumbs. Okay, so next we'll be moving into the next part, or this is the second important component of DNA. Okay, so next if you are looking into the next component, the third component, you have to keep in remember that it is third component is always a phosphate group. Okay, always it will be a phosphate group. Okay, so if you are considering phosphate group, this phosphate will be attached to 4 oxygen molecules. Okay, if one set of phosphate group is attached, then we will call it as monophosphate. Okay, if 2 set is attached, then we will call it as diphosphate. If 3 set is attached, then we will call it as triphosphate. Okay, so monophosphates will be present. Only one set of phosphate group are attached, then we will call it as monophosphate. If 2 sets, then we will call it as diphosphate. If 3 phosphate groups are attached, then we will call it as triphosphate. Okay, so for example, I'll say a thing that is ATP. Okay, adenosine triphosphate. This is important thing or important molecule that you are studying for your respiratory activity, right? ATP is produced, ATP is synthesized, ATP is synthesized. What is actually ATP? It is adenosine triphosphate, right? Adenosine, what is adenosine triphosphate? Okay, so see adenosine means adenine is attached, ribose sugar is attached with adenosine. Okay, adenine, then we will call it as adenosine. Then triphosphate, if it is attached with a 3-phosphate group, then we will call it as adenosine triphosphate. Okay, clear. So you have to connect everything with this. Okay, then if you are considering ATP, then you might be knowing ADP too. Okay, it is adenosine diphosphate. Okay, if 2-phosphate is attaching, then we will call it as adenosine diphosphate. Okay, next one, if you are again considering AMP. Okay, AMP, adenosine monophosphate it is. Okay, adenosine monophosphate. So these are the 3 different types of molecules that can be present. Okay, in the case of DNA, this AMP is formed. Okay, in case of see, adenine is a kind of base pair. So if adenine is formed with this monophosphate, then we will call it as adenosine monophosphate. We'll come to learn that in detail in the coming slides. Okay, so the third important component. What is the third important component? It is phosphate group. So you got the 3 different components of DNA, right? What are the 3 different components? Once again, if you are considering the 3 different components, first one it is ribose sugar. Okay, so if you are considering DNA and RNA, in DNA we will call it as deoxyribose. Why it is known as deoxyribose? The second carbon, if in the case of second carbon, what you can consider? The second carbon, the oxygen group will be eliminated. So, we will call it as deoxyribose. But if in the case of RNA, that oxygen will be present and it can act as a nucleophile also. Okay. So, next one, if you are considering the second component as, the second component will be nitrogen bases. Okay, what are the 4 different nitrogen bases? They are purines and pyrimidines. In purines, it is adenine and guanine. In pyrimidines, it is thymine and cytosine. Whereas in the case of RNA, the thymine will be replaced by uracil. Okay, so these are the nitrogen base. So next it will be considered as the phosphate group. Now we have to know how this is combined together. Okay, or how these components are combined together to form a structure of DNA. Okay, that is also important, right? So if you are preparing something for your curry, we'll cut it and we'll separate it and we'll put it away, put it aside, right? Later on, we'll mix it together to take or to make it a curry. Okay, so exactly same like that. We have different components here. Okay, so what we want to make? We want to make it as a complete entire DNA. Okay, so let's see how this DNA is formed. Okay, so we have ribose sugar, right? What we have? One component as ribose sugar. Okay, so this ribose sugar will be attached to ribose sugar is present, and this ribose sugar will be attached to the nitrogen bases. Okay, this ribose sugar will be attached to the nitrogen bases, and this, if it is attached to the nitrogen bases, then the structures will be known as nucleosides. Okay, it will be known as nucleosides. Dear students, it is very important what they are known as nucleosides. Okay, nucleosides. For example, I'll be writing one example here. So it will be easy for you to understand. Okay, so let's draw the structure of ribose sugar here. Okay, this is a ribose sugar. Okay, this is a ribose sugar. So first position OH is present. HOH, then H. Okay, here there is another H, then HOH. Then the fourth position, if you are considering, H is present and CH2OH is present. Okay, this is the structure of a deoxyribose sugar. Okay, not ribose here. In the case of RNA, it will be ribose sugar. In the case of DNA, it will be deoxyribose. Okay, deoxyribose. Okay, so here I have drawn the deoxyribose sugar. So in the first position, you can find an oxygen. In the third position, you can find the oxygen. And in the fifth position also, you can find the oxygen group. Okay, so if oxygen group is actually present, it has the ability to attack the other molecules, or it has the ability to share the molecules. We can say they have a property of nucleophile. Okay, so nucleophile attack will be present, or nucleophilic attraction will be present. So with this nucleophilic attraction, this first group will be attached to the nitrogen bases. Okay, to the first group, what it will be attached? To the first group, what it will be attached? With, if you are considering, this OH will be moved or removed. Okay, this OH will be removed. Instead of OH, what will be present? The first group will be attached to the nitrogen base. Okay, so there is a nitrogen base here, and that nitrogen base is adenine. Okay, clear? So the first carbon, if you are considering the first carbon is attached with the nitrogen base. This is the first carbon, this is the second carbon, this is the third carbon, and this is the fourth carbon, whereas this is the fifth carbon. Okay, so we can write the first carbon. Okay, the first carbon is attached with nitrogen base. Okay, first carbon is attached with nitrogen base. So there is a bond actually formed in between, right? And the bond formed here, is N-glycosidic bond. Okay, N-glycosidic linkage or N-glycosidic bond. It is very important. See, it is N-glycosidic bond. When you're studying more about this, you will come to know that there are another type of glycosidic bond too, not only N. So here it is clearly mentioned that it is N-glycosidic linkage. Okay, what linkage it is? N-glycosidic linkage. So you can make sure one word question will be asked from this section. Okay, first carbon is attached with the nitrogen bases. With which bond? It is N-glycosidic linkage. Okay, N-glycosidic linkage or N-glycosidic bond. Okay, likewise, we can write every nitrogen base. Okay, if it is here, if adenine is not present, if thymine is present, then it is known as another nucleoside. Okay, so different nucleosides can be present, and we will call it as, if adenine is present, we will call it as deoxyadenos. Okay, we'll call it as deoxyadenos. If thymine is present, we will call it as deoxythymidine. I'll show the nucleoside, the names of nucleosides are written here. Okay, if adenine is present as the nitrogen base, the nucleoside formed will be known as adenosine in case of RNA. So here we can write, or here I can write it as, these are the components of RNA. Okay, these are the components of DNA. See, adenosine is present in RNA. Okay, deoxyadenosine is present in DNA. Okay, so if you're considering guanosine is present in RNA. Deoxyguanosine is present in DNA. Thymidine is present in RNA, whereas deoxythymidine is present in DNA. So then cytidine is present in RNA. Deoxycytidine is the component of DNA. Uridine is the component of RNA, whereas deoxyuridine is the component of DNA. Clear? So these are the nucleoside. Okay, these are the different nucleoside, possible nucleosides that is formed in DNA. Very important. Dear students, it is very important to study the names. Okay, study the names. Make a note on it. Okay, so these are the nucleoside. So you understand how the nucleoside is forming, right? So next we have to learn how nucleotide is formed. Okay, how phosphate, the third component is phosphate, right? So how phosphate will be attached to the nucleoside. So next we will be studying about nucleotide formation. Okay, nucleotide formation. So let's see. So with adenine, I have told you, I have mentioned how to draw. Okay, so first position it will be HOH, and then the fourth carbon is attached to the hydrogen. Then finally it is CH2OH. Okay, so in the first position, what is present? Nitrogen base is present, and this is known as nucleoside. Okay, and this is known as nucleoside, and we can write adenine as the nucleoside here. Okay, adenine as the base pair here. Okay, it is attached to the base pair, ribose sugar with the help of which bond? N-glycosidic bond. So next you have the phosphate group. The phosphate group also has to get attached with this, right? So the phosphate group will be attached to the fifth carbon. Okay, phosphate group will be attached to the fifth carbon. Okay, which carbon? It will be getting attached to the fifth carbon. See, CH2 is present already here. OH, by nucleophilic reaction, it will be moved and phosphate group will be added. Okay, so you have to again mention which linkage it is. See, this is the first carbon. Okay, this is the first carbon. This is the first carbon. This is the second carbon. This is the third, and this is the fourth. This is the fifth. Towards the fifth carbon, what is attached? Phosphate group is attached. Okay, so when I was saying about this nitrogen base was attached to the first carbon with N-glycosidic linkage, right? So the phosphate group is attached to the fifth carbon with the help of phosphoester linkage. Okay, which linkage it is? Phosphoester linkage. Okay, so we can say the phosphate group. Okay, the phosphate group is attached. Okay, the phosphate group is attached to the fifth carbon. Phosphate group is attached to the fifth carbon. Through which bond? It is through phosphoester linkage. Okay, clear? When phosphoester linkage or when phosphate group is attached to this, then we will call it as a nucleotide. We will call this as a nucleotide. nucleotide. Okay, what we will call nucleotide. Okay, so how understood you? Did you understand how nucleotide are formed? Okay, so if nucleoside is present, towards this nucleoside, what is added? Phosphate group is added. Phosphate group is added to the fifth carbon through which bond? It is through phosphoester linkage. Okay, clear? So if adenine is present, it is formed as AMP. See here, only one phosphate is attached. If two phosphate groups are attached, then it will be known as ADP. Okay, adenosine diphosphate. If three phosphate group is attached, then we will call it as adenosine triphosphate. I told you adenosine triphosphate is the molecule that is being produced inside the mitochondria after the cellular respiration. Right. So that molecule is very much important since it is an energy molecule. It is important. It is also important in the synthesis of DNA. Okay, so if adenosine triphosphate is cleaved into adenosine monophosphate, it can be utilized for the formation of DNA. Okay, clear? So, see, AMP is formed. Next, if guanine is formed, guanine is present instead of adenine here, then we will call it as GMP. Okay, if biphosphate is formed, then we will call it as GDP. Then triphosphate is formed, then we will call it as GTP. Okay, so next one, it is cytosine, right? If cytosine is present, we will call it as CMP. Okay, if 2-phosphate is formed, then we will call it as CTP. Then if 3-phosphate is present, then it will call it as CTP. Okay, so next one, we have to know about thymine, right? If thymine, then we will call it as 1-phosphate, then we will call it as thymine monophosphate. Okay, if 2-phosphate is present, we will call it as thymine diphosphate. Okay, what we will call? We will call it as thymine diphosphate. Okay, if 3-phosphate group is present, we will call it as thymine triphosphate. Okay, so if you are considering TMP, thymine, then it will be forming TMP, monophosphate, then PDP and TTT. Then next, if you are considering next one, it will be UMP. Okay, uridine monophosphate. Then we have to know about uridine diphosphate and uridine triphosphate. Okay, so among this, I didn't write about ADP and ATP. Okay, these are known as nucleotide. Okay, nucleotide. Dear students, it is not necessary to know about ATP, GTP, CTP or TTP or UTP now. But if you are considering the next chapters, if you are considering the polymerase chain reaction, okay, you will come to know about this in the coming chapters. Okay, I think it is in biotechnology chapter. So when we are dealing with that, we have to add some nucleotides into that. Okay, so when we will be using the term, then at that particular point, we'll be using the term as UTP and GTP, TTP, etc. So it is easy to understand what is TTP, then UTP, ATP, GTP and CTP. Okay, fine. So you got what is nucleotide, right? What is nucleotide? If a ribose sugar is attached with a phosphate group, then we will call it as a nucleotide. Okay, so that's the components of how the components have been arranged. So here you have one nucleotide, right? Here you have one nucleotide. Then we have to know how another nucleotide will be getting attached to the one nucleotide. Okay, so I'll be drawing again here the nucleotides. Okay, fine. So if you're considering the nucleotides, this is the first nucleotide. Okay, here you have a nitrogen base. Okay, then HH is present. Then here HOH is present. Then here you have the phosphate group CH2. Then PO4, right? Okay, this is the first group. So this is the first nitrogen base. Okay, so sorry, this is the first nucleotide. So we can write here as adenine. So next nucleotide, another nucleotide is present. Okay, so we can write the another nucleotide here. Okay, so this is the second nitrogen base. Okay, so here we have CH2, PO. Okay, so this is the second nucleotide present. How two nucleotides will be combined together? Now that is the question. See, we have one nucleotide, we have four nucleotides present in DNA, right? So here we will be writing timing. Okay, so how these nucleotides will be attached together? See, this is the first nucleotide and this is the second nucleotide. Okay, if you're considering this is the first nucleotide and this is the second nucleotide. Okay, so the first nucleotide, we will write the numbers too. Then first carbon is this and second carbon is this, third carbon, fourth carbon and this is the fifth carbon. Okay, if we are considering here first carbon, second carbon, third carbon, fourth carbon. Sorry, this is the fourth carbon and this will be the fifth carbon. Okay, so here how the attachment, how nucleotide, two nucleotide will be combining together. See, the third position you can find a free O here, right? So it will be acting as a nucleophile. And again, the nucleophilic reaction will be taking place and this hydrogen will be removed. Okay, this hydrogen and oxygen will be removed. So this bonding will be like, okay, it will be removed. So the third carbon is attached. The third carbon is attached to the phosphate group. Okay, the third carbon is attached to the phosphate group through which bond here there is the bond present here is phospho diester bond. Okay, what bond is phosphodiester linkage or phosphodiester bond. Okay, so this is how two different nucleotides are being attached. That means third position OH is present and this third position OH is attached to the fifth carbon of the second nucleotide. Okay, fifth carbon, there you have phosphate group. So it will be attacking to the phosphate group of the fifth carbon. Okay, so through which bond it is attacking? It is through phosphodiester linkage. So we have learned how different things have been formed. Okay, we have learned how nucleoside is formed, how nucleotide is formed, how two nucleotides will be combined together. So we'll write the linkage all together once again. So you can make a note on this. Okay, so first one, if you're considering, you have studied the nitrogen base, right? Nitrogen base is attached to the ribose sugar. Okay, ribose sugar through which bond? Through which bond? Through which bond? It is through N-glycosidic linkage. Okay, it is through N-glycosidic linkage. Okay, next if you are considering, okay, second one. First one, this one. Second one is phosphate group. Okay, phosphate group is attached. Phosphate group is attached to a nucleoside. Okay, to a nucleoside. Then how the bond will be? What is the bond there? What is the bond present there? It is phosphoester linkage. Okay, it is phosphoester linkage. Okay, it is phosphoester linkage. So next one, third one you have learned about. Okay, third one you have learned about phosphodiester linkage. Two different nucleotides. Okay, two different nucleotides. Two different nucleotides are connected by which bond? It is connected by phosphodiester linkage. Okay, phosphodiester linkage. So this is very important. You can expect at least one MCQ from this section. Okay, one MCQ from this section. So be thorough with this. One more bond we have to learn. We will be learning now. Okay, so this is about how two nucleotides are formed. See, I have right here about nucleotide. It will be monophosphate, diphosphate, or triphosphate. So this is the structure of a polynucleotide. See, first you can find the first nitrogen or first nucleoside here. Okay, the second nucleoside here. The second nucleoside is this. Third nucleoside is this. Fourth one is this. Okay, when phosphate group is attached and it is connected, it becomes a nucleotide and it becomes a polynucleotide. Four different base pairs are present here, right? So adenine is present, thymine is present, guanine is present, and cytosine is present. So we can consider this as a polynucleotide. Why polynucleotide? Poly means many. Okay, so it will be known as polynucleotide. Okay, and these polynucleotides are connected each other with the help of which bond? It is by phosphodiester bond. Next, you have to study. See, if you are considering the first carbon, dear student, this is also very important. Okay, when you are studying about replication, you will come to know 5 prime, 3 prime position. So, what is actually there in 5 prime and 3 prime position? Okay, or why it is known as 5 prime and 3 prime position? So, let's see. Okay, so if you are considering, this is fifth group, right? This is the fifth carbon and fifth carbon is attached to a free phosphate here. Okay, so if you are considering this end position, a free hydroxyl OH group is present. Okay, so the position where free phosphate is present will consider as 5 prime position. Okay, we'll consider as 5 prime position. It's a simple logic. It is fifth carbon. Towards fifth carbon, what we are attaching? Phosphate group is attaching. So, where the 5 prime or fifth phosphate group is present, we will call it as 5 prime position. Okay. So, you know that in the third position of your ribose sugar, what is present? Hydroxyl group is present. Right. So, that is why we will call this region as 3 prime hydroxyl region, or we will just mention it as 3 prime. Understand that if 5 prime is present, then free phosphate group is there. If 3 prime is present, then free OH group is there. Okay, so clear? Clear? So next we are moving into the structure of DNA. How DNA is formed? Okay, how DNA is formed. See, this is the structure of polynucleotide. I told you that is one polynucleotide. Thus, if two polynucleotides are present, this is the first polynucleotide and this is the second polynucleotide. Okay, this first polynucleotide and the second polynucleotide are arranged anti-parallelly. Okay, not parallelly, they are arranged anti-parallel. Okay, anti-parallel. Okay, anti-parallel. Okay, so they are arranged anti-parallelly. So see, first carbon, first polynucleotide, if you are considering the position, see this one is 5 prime position. Okay, this one is in 5 prime position and this is in 3 prime position. Okay, this is in 3 prime position. First one is 5 prime position and the second one is in 3 prime position. Okay, so what are the different base pairs present here? It is adenine, thymine, guanine and cytosine. If you are considering the next one, adenine will be pairing with thymine. Thymine will be always pairing with adenine. Guanine is pairing with cytosine. Cytosine is pairing with guanine. Okay, see that pairing you can find. Okay, so next one, if you are considering next polynucleotide, see where is the 3' position. Okay, you can find a 3 prime position here and 5 prime position here. That means they are anti-parallel. Okay, that means they are anti-parallel. Okay, clear? Okay, so if you are considering the structure of DNA, they are anti-parallel. And always think about Chargaff rule. Always think about Chargaff rule that adenine always pairs with thymine. Okay, adenine always pairs with, say, adenine always pairs with thymine. Okay, guanine will always pairs with cytosine. Okay, guanine will be always pairing with cytosine. Adenine is always pairing with thymine. Guanine is always pairing with cytosine. Also, which bond is present here? Which bond is present here? Hydrogen bond. Okay, what is the bond here? Hydrogen bond is present here. Okay, so in between adenine and thymine, which linkage or which bond is making them together? It is hydrogen bond. If you are considering adenine and thymine, how many hydrogen bonds are present? How many hydrogen bonds are present? Two hydrogen bonds are present. Okay, if you are considering guanine and cytosine, how many? Three hydrogen bonds are present. Okay, so for a simple technique to remember the number of bonds, C is the third letter, right? C is the third letter in alphabetic order, right? A, B, C. So if cytosine is present, it will have three bond with guanine. Okay, if you study one, the other will be automatically you will come to know that in mind automatically. Okay, so adenine always pairs with thymine, guanine always pairs with cytosine. That means if amount of DNA, if amount of adenine is present, for example, we can consider 20%. Okay, adenine is 20%. Adenine is 20% in a DNA. So we can consider that thymine in the DNA will be always 20%. Okay, the amount of adenine and thymine will be equal. So if adenine and thymine is known, then we can come to know about guanine and cytosine, right? If adenine and thymine, then we can add this as A plus T, and A plus T is equal to 40%. So we can find what is G plus C. Okay, what is G plus C? It is 100 minus 40. That is 60. Okay, that is 60. If guanine will be is equal to 60 by 2, that is 30%. Okay, if guanine is 30%, then cytosine will be 30%. It will be same. Okay, so if you are asking, if the question is asked to write the amount of purines, then you can add adenine and guanine. That is 40 plus 30. It will be, sorry, not 40, 20 plus 30, it will be 50%. Okay, clear? This is very important. You have to be thorough with this equation and also about this Chargaff rule. Okay, next it is about central dogma. This is very important, very important. Repeatedly, any question is based on this one particular small diagram. Okay, see what is central dogma? See, there is DNA present, right? We are saying about DNA. We are considering about DNA. Okay, so if DNA is present, from the DNA, again the DNA will be forming. Okay, that is known as replication. Okay, that is known as replication. So if you are considering DNA formation from DNA, then we will call it as replication. The next thing we have to learn is about replication. Okay, so this DNA is converted, or this from this DNA what is formed? mRNA is formed. Okay, and this mRNA is formed with the help of a process called transcription. Okay, what the process is known as? The process is known as transcription. Okay, from mRNA, what is formed? Protein is formed. Okay, from mRNA, what is formed? Protein is formed, and the process is known as translation. Okay, clear? So it is very important. Study. What is central dogma? Okay, make sure that you understand and you practice how to write this. Okay, fine. So let's see these DNA. You have studied the polynucleotide. You have studied how two different polynucleotides will be attached, right? Adenine will be pairing with thymine. Thymine will be pairing with adenine. Guanine will be pairing with cytosine. Cytosine will be pairing with thymine. Okay, so see, not cytosine will be pairing with guanine. Okay, so these things are done to form two different or to attach two different polynucleotide, and you have to understand that the structure of DNA is anti-parallel. There is double-stranded DNA. Okay, DNA is always double-stranded DNA. That is why we are attaching one polynucleotide with the other polynucleotide with the help of different nitrogen bases. And here, what you have? Oh, you will have hydrogen bonds. Okay, it's the condition. So let's see how this DNA is being packed inside the nucleus. Okay, packed inside the nucleus. So if you are considering the distance between two consecutive base pairs, it is always 0.34 nanometer. Okay, so if you are considering one base pair here, and the second base pair here, third base pair, fourth base pair, the distance between these base pairs will be always 0.34 nanometer. Or else we can write it as 0.34 into 10 raised to minus 9 meters. Okay, so what is the length of a DNA? If you are considering the length of DNA is considered as the distance between two consecutive base pairs and the total number of base pairs present. Okay, the total number of base pairs present in DNA is around 6.6 into 10 raised to 9. Okay, so if you are multiplying this 0.34 into 10 raised to minus 9 with 6.6 into 10 raised to 9, then we will get the answer as around 2.2 meters. Okay, this calculation is very important. You have to keep it remember. Okay, so what is the length of a DNA? What is the length of a DNA? If you are considering the length of DNA, the number of base pairs present in it is multiplied with the distance between the two consecutive base pairs. Okay, so now how the DNA is being wrapped inside, or how the DNA is being coiled, or see, if you are considering the polynucleotide, it is long, right? It is very long. Okay, this is the double-stranded DNA. This is the double-stranded DNA. It is, as I have mentioned, it is 2.2 meter. It is 2.2 meter. In every nucleus, see, in every cell, what is present? In every cell, what is present? What is present? The nucleus will be present. Okay, in every cell there is nucleus, and inside this nucleus what has to be done? This 2.2 meter long lengthy DNA has to be condensed. Okay, so now how this condensation is formed, or how this condensation is done, that is given here. Okay, so how is it possible? How is it possible? This long DNA is wrapped around. Okay, see, this is negatively charged DNA. Okay, what is this? This is negatively charged DNA. This is negatively charged DNA, and this negatively charged DNA is wrapped around a protein molecule. Okay, it is wrapped around a protein molecule, and that protein molecule is known as histone protein. What protein is known? It is histone protein. So, if you are considering the histone proteins, they are actually octamers. Okay, they are actually octamers. Okay, they are actually octamers. Octamers means they have 8 different subunits. Okay, 8 subunits where 4 each. If we are considering the octamers, they are containing H2A, then H2B, H3 and H4. Okay, so this units will be present as twice. Okay, so we'll call it as octamers. These octamers are connected with a linker protein. Okay, are connected with a linker protein, and this linker protein is known as H1 histone. Okay, linker protein is known as H1 histone. So, this DNA, at first this DNA will be wrapped around a histone octamer, and that particular structure is known as nucleosome. Okay, what is the structure known as? The structure is known as nucleosome. How many base pairs will be present inside the nucleosome? 200 base pairs will be present in each nucleosome. Okay, if you are considering the number of base pairs, how many base pairs will be present? 200 base pairs will be present. Okay, then this nucleosome will be again condensed to form chromatin fibers. Okay, it will be again condensed to form chromatin fibers. Before that, they will form a structure known as beads on a string. Okay, they will form a structure known as beads on a string. Okay, if you are considering this as together, then it is known as beads on a string. Later on, it will be forming a chromatin structure. Okay, then after that, during metaphase, it will be forming a structure known as chromosome. Okay, you know that during cell division, there is mitosis, right? Mitosis taking place. So, when mitosis is done, then during S phase, synthesis phase, it will be transformed into a chromosome structure. Okay, clear? So, this is how the DNA is wrapped or coiled, highly coiled inside the nucleus. Okay, so all right, you understand? I think you got this, right? So, what we have learned today? What we have learned today? First, we have gone through the structure of DNA, right? How the DNA and RNA can be made. It can be made with the three different components, that is ribose sugar, then ribose sugar is there, nitrogen bases are there, then what is there? Phosphate group is present. Okay. With the three, what can be formed? DNA and RNA can be formed. If you are considering DNA, the sugar, ribose sugar will be deoxyribose. Okay. If you are considering RNA, the nitrogen base is present instead of thymine, it will be uracil. Okay. Done. So, this is how nucleoside is formed. From nucleoside, a phosphate group is attached, then nucleotide is formed. From nucleotide, what is formed? Polynucleotide is formed. Polynucleotide, the DNA is formed. Okay, so that is how the DNA is forming. Then from the DNA, you have to study how the DNA is being placed inside a small nucleus. The 2.2 meter lengthy DNA is placed inside a small rounded nucleus. Okay, it is highly wrapped into the structure known as chromosome. First, it is wrapped into a positive protein known as histone proteins. Okay, so from there, the structure is a new structure is formed, that is nucleosome. Nucleosome will be forming a beads on string structure. Then chromatin is formed. From there, chromosomes are formed. Okay, so one more thing is there. Not only the histone proteins, another non-histone proteins are also helping in the formation of chromosomes. Okay, with that, you have to learn about two different terms too. That is about euchromatin and heterochromatin. Okay, euchromatin and heterochromatin. Okay, so if you are considering euchromatin, this euchromatin are loosely packed DNA it is. Okay, loosely packed DNA is known as euchromatin. Okay, so it will be staining less. Okay, stains less, and also they are transcriptionally active. Okay, they are transcriptionally active. Okay, so if you are considering heterochromatin, they are tightly packed. They are tightly packed chromosomes. Okay, they are tightly packed chromosome and they stain more. Okay, and also they are transcriptionally inactive. Okay, so this both euchromatin and heterochromatin will be present inside the chromosome. Okay, transcriptionally inactive DNA. Okay, transcriptionally active and transcriptionally inactive means transcriptionally active chromosome will undergo the process known as transcription, formation of mRNA. Okay, transcriptionally inactive means that DNA won't undergo the transcription process, that the process of mRNA and later on the process of protein. From that particular part, no protein will be synthesized. Okay, clear? So, this is the end of our session. So we'll be dealing with the rest of the portion in the next class. Okay, in the next class, we will be discussing how replication, what are the different experiments on replication. Just go through and come for the session. So it will be easy for you to understand. Okay, so thank you. So Dania ma'am is again waiting for you for Saturday on 28th October 2023. She will be again coming with solutions. Okay, part 2 it will be. So wait for her. Be in time. Thank you.